Welcome to today's webinar on Introduction to Data Visualization using R for biologists. So in this webinar, I'll be introducing you to the basics of data visualization, and I'll also demonstrate on how to use R and R Studios for creating visually appealing graphs for your data, right? So when we talk about data visualization, right, it's important to understand the evolution, its evolution over the time. So if you look at the first graph here in the left, so this is basically one of the earliest examples of data visualization is a graph, which dates back over 800 years ago, where astronomers or mathematicians, it's not clear who, but whoever they are, they have tried to use charts or diagrams to illustrate the position and trajectory of celestial bodies over the movement of celestial bodies over time, right? So if you look fast forward to 200 years ago, the graph that you see in the right, this is a graph, it's a line chart to compare exports from England with imports into England in the year 1700 to 1780. Now this graph is created by somebody called William Playfair. Okay, so William Playfair is was a Scottish engineer, and he has made several contributions to the field. So he has been given the credit for many graphs that we know today, like line charts, bar charts, and even pie charts, right? So since then, the data visualization has evolved significantly, and with the emergence of computers and software like R and Python, now we can create much more sophisticated graphs, way more easier, and it's easily accessible as well, right? So, sorry. Right. So before we start, before we go further, it's important for us to know what exactly data visualization is. So in simple term, it is nothing but the graphical representation of data. So as you can see here in this flow chart, it is usually starting with a data set. And this particular data set is, processed and analyzed by data analyst or researcher, where they try to generate different types of graphs to understand the data. And they, once these different types of data visualizations are created, these graphs are then used further to communicate the insights to a large range of audience. It could be policymakers, stakeholders, or even to general public. So as they say, a picture is worth 1,000 words. So it's easier for one to explain or even convey a certain message using a graph or a visual representation, right? So now we know what this data visualization is. Now let's look at why it is important, right? So, for starters, let's say our brain is wired in such a way that visual informations are processed, right, is processed much faster than text and numbers, okay? So by presenting the data in a visual format, we can quickly identify the outliers, correlations, or any other insights that might be harder to spot in a table or an Excel sheet, right? So here is an example of an Excel sheet where this is a demo data of, if you look at the A column, you have multiple genes, gene 1 to 20, and the rows, you have multiple samples. So it's a demo gene expression data. But just by looking at this particular data, it is difficult for us to come up with any correlations or identify any pattern. It is quite difficult. But the same data, when I'm trying to make it into a heat map, So let's not really, we don't have to worry about what heat map is right now. Heat map is nothing but, graphical representation which is used to show the correlation between two variables. Okay, so in this, you can easily spot some relations such as, for example, sample 1 and gene 3, there is a positive z-score. Similarly, if you look at sample 4 and gene 12 here, there is a negative z-score. So let's not worry about what z-scores are. This is just an example how visual representation can make the, identifying patterns way more easier. Okay, so moving to the next example. Okay, so let's move forward. Let's take a look at the different types of data visualization. So broadly speaking, there are two main types of data visualization. One is exploratory and the other one is explanatory. So exploratory data visualization is used to explore the data and generate insights. So it's typically done by people who are not, who don't understand the data much. So if you have a new data and we are not sure what we are looking for, we first explore the data. So exploratory data visualization involves creating multiple types of graphs, okay? And from those graphs, we try to understand if there are any patterns or, any correlations, right? So like the name suggests, exploratory data visualization, it is for exploring the data. On the other hand, explanatory data visualization is used to communicate insights. So let's say this type of visualization is typically more structured and targeted. Okay, so it often involves in creating a single high impact chart or a graph that conveys the key message in a way, in a clear and compelling way. Okay, so now that we understand what data visualization is and why it's important, let's talk about how to choose the right type of graph for our data. All right. Okay. So nowadays, there are many types of graphs and charts available to visualize data, right? And it's very important to choose the right one to effectively communicate your message. Selecting the wrong type of graph can not only make it difficult for your audience to understand, but it can sometimes mislead them to, draw incorrect conclusions as well. So here you see there are several types of graphs, right? Bar chart, line chart. Some of these graphs are something that we have come across at least once in our life, right? So how to choose the right one for your data? So the first point will be looking at your data itself. So are you working with a categorical or a numerical data is first thing that you need to identify. So there are different types of data. However, here we will just be talking about the broad categories that are categorical and numerical, right? So categorical data is basically contains data points which cannot be measured on a numerical scale, right? For example, gender, colors, and education level. So here there's an example you can see. Yeah. So we have, there's a survey conducted in a school, right? So you have in the y-axis the number of children who have chose the colors. So this is just a bar graph showing the favorite color of students in preschool, primary, and secondary. So if you look at the x-axis, it belongs to different categories, right? So these categories cannot be really measured or converted into numerical scale. So we know that we have a categorical data, right? If you look at some the other example, such as age, income, or height, these are some of the data that you can measure using a scale, right? Numerical scale, their respective numerical scale. So these kind of data is classified into numerical data. So for numerical data, if here there's an example, in the y-axis, you can see the ear length by height. And the x-axis, you can see the age. All right. So here, the difference, if you look carefully in these two graphs, you can see in the numerical data graph, right, the right graph, both the x and y axis are numerical scale, right? So it's the first point is to identify the type of data you have. Once we have done that, we will then determine the purpose. So what are you trying to, why are you making the graph? What are you trying to tell the audience? Are you trying to compare values, or are you trying to show a trend or demonstrate a distribution? So the second point is the purpose. So once we have decided what type of data you have, second would be to determine the purpose, right? So for example, I have a categorical data and I want to show frequency distribution like how I showed here, sorry. Here, right? So this is a categorical data and I wanted to show the distribution. So I have used a bar chart here, right? So you can use either a bar chart or a pie chart. So this is a table that shows you the recommended graphs for different type based on the data type and purpose. So these are the two major, pointers that you should remember when you're choosing a graph for your analysis, right? So creating effective visualizations involves more than just choosing the right type of graph, right? It's also important to consider how the graph is designed and presented. So in this coming section, we'll discuss some key design principles that can help make your visualizations more effective, okay? So let's go one by one. First, so here, the first point would be, again, importance of choosing. So earlier we discussed how to choose the right type of graph, right? So here I'll be telling you why it is important to choose the right type of graph. So here I have a data of government budget for different government agencies in US, okay? And they have plotted this particular data in a pie chart. There are several things that are wrong here. For example, they have used colors, so many bright colors, and they have used multiple shades of orange, okay? Which makes it a little difficult to differentiate. Second, there are several slices or pie slices they call on the top left here. I'm not sure if you can see my pointer, but in the top left, you can see several small slices, and it's again very difficult for me to differentiate between them. The same data, I can make it into a bar chart, which can give me a clearer information, right? So again, choosing the right type of graphs really matters, okay? Let's move to the second point. Right. So here I have a graph. It's a bar graph, okay, which shows number of deaths per month in Iraq from the year 2003 to 2011. Right. So this visualization is created by Simon Scar. Okay. So if I look at this graph, I can see there's an increased number of deaths in the middle around some time, and then there is again a decrease, right? These are some of the conclusions that I can come up with. However, look at this. What do you see? The first thing I see is a smear of blood dripping down the screen, right? So both the charts are basically telling the same fact, but the way of representation makes a different impact on the viewers. And the right side graph is way more it captures your attention, right? So you could, you could just see how easy it is to control the message, right? Only by changing certain parameters, right? So the three simple choices that the creator made here was one, changing the title, second, the color of the graph, third, the orientation of the bars, okay? So yes, so this is one fine example on how using color and design effectively can make a difference, right? Moving on. Okay, so here you can see a bar chart showing the sales in the company over time, right? So certain things I can notice here is excessive, data labels. There are so many numbers here. There's a lot of annotation that is present. And overall, I could feel that this particular graph is quite crowded. Okay, and this can be avoided. Sometimes what it does is it would distract the audience from the real message. So it is very important to avoid clutter and unnecessary elements from your graph. Okay, so moving on to the fourth point. Okay, so let's look at this graph here. So this is a bar chart showcasing the gross margin percentage of net sales, right? So if you look here, I can see there's a reduction in sales in the year 2016 October here, right? And there's again a spike in the November, right? So there are certain conclusions I can make it. The data is highly volatile, but if you look carefully here, look at the y-axis. It starts from 34. Conventionally, your y-axis should start from 0, right? So this could actually lead to mislead you, basically. And if you look at the actual data, where the y-axis starts from 0, it tells a different story. So using appropriate scales is again one of the very important aspects when you are creating or making a graph. Right. Fifth and one of the most important point is knowing your audience. So if you look at this particular graph, you can see the quantitative treatment efficacy of drugs, right? So you can see tumor volume, and then you can see the time duration. There is other data like survival rate, etc. But if you're showing this data to a scientist or a doctor or somebody who has a prior knowledge about how drugs work, what tumor volume is, right? This would make sense. But if you're just presenting this particular graph to somebody, maybe patients or a wider or a larger, audience, this would not be the right representation. Let's look at the other part. Okay. So the graph in the right side is more simple, right? It's easy to understand by somebody, even if that person does not have much scientific knowledge. It is easy for that person to understand it. So knowing your audience before you create a graph is again one of the most important principles, right? Okay. All right. So it's important to remember that not all graphs tells you the truth. Okay, so some may be misleading either intentionally or unintentionally. So as a researcher or a data analyst, it's important to be aware of these pitfalls, right? And avoid them as whenever possible. So I'm going to be showing you some of the examples where data visualization or graphs can be used to mislead you, right? Okay. So the first impression that I get from looking at this particular data is, oh my God, the interest rates are, skyrocketing, right? But again, remember, look at the y-axis. It's starting from 3.140, not starting from 0, right? Let's see how it looks when, yeah. So it makes a difference, right? So if somebody had to mislead you, they would use the first graph. So always, whenever you are trying to draw any insights or conclusions from a graph, look for the y-axis, okay, and see where it's starting from, okay? Next, all right. So here, so inconsistent or manipulated scale. If you look at this graph here, it talks about the cost of sugar. Okay, cost of sugar, and over the time, over the time of 6 months, I guess. Yeah. So when I'm looking at this graph, I can see that, the cost of sugar pretty much remains constant. It's not much fluctuating, right? However, if I further expand the, y-axis, I can see that it is fluctuating, but, it's not fluctuating much, but it is, right? So compressing or expanding the scale of the graph can actually make changes between the data points, right? So always look at the X and Y axis and then draw the conclusions from the graph. Okay, interesting. Cherry picking. So cherry picking is basically, it refers to the practice of selectively choosing data to represent. Okay, so it means that the creator or the presenter has omitted a lot of data and decided to show you only a small subset of data. Okay, so here looking at this particular graph, I can say Canada is doing a great job. Its gasoline prices are, decreasing. Yeah, its gasoline prices are decreasing. So this data, so they try to make this graph look like this by selecting only a certain time frame, right? May 2019 to February 2020. However, if you look at the whole picture from May 2019 to November 2021, it's decreasing, but it's also increasing, right? So it's important to avoid the cherry picking or subsetting your data for presentation, and present the data in more, let's say, unbiased or more transparent manner. So whenever somebody is showing you a graph, the first graph, let's say, always try to question what about the data after February 2020. Okay, you never know what you would find out. Okay, choosing the right colors. So here you have the map of US, and here they have, they are showing the reported cases of coronavirus. It's long back, okay? Reported cases of coronavirus. So the first impression that I get from looking at this particular data is the red ones are highly infected, right? That would be the conclusion that normally anybody would go. The greens are safer, the red ones are dangerous, right? So if you look at the scales, it's not really true. Here it says red ones are 1,001 to 5,000, and the black ones are 5,000 to 10,000, and the light green ones are 10,000 or more. So you see how whenever we use colors in unconventional way could actually give a wrong information to the audience, right? So this is one example for using colors in unconventional way, right? And the second graph in the right, it's a line graph. They have used multiple shades of orange, red, and pink. However, it's very difficult to differentiate between them, right? So whenever we use colors, it's important to make sure that we use contrasting colors or colors that you could differentiate. So when used thoughtfully, color can make it easier to spot the trends, right? However, color can also cause confusions. So be careful about the colors that you choose for your representation, for your graphs. Okay. So now here, I would like to touch upon a concept called correlation and causation. Okay. So if you look at this graph in the first, I can see cigarette smoked and then lung cancer death. Both the line graphs are almost similar, right? So I can draw a conclusion that it's correlated. So can I draw a conclusion that it is causation by just looking at the graph? No. Okay. However, you need enough, there have been enough research done in the backend, enough data analysis that is done to establish the link between, smoking and cancer. So today we know smoking and cancer is related, and smoking causes cancer, right? So here you can see the correlation as well as causation. The causation part only because there's a research that supports this. Now let's look at the second graph. where they're talking about ice cream sales and shark attacks. You see how both the lines are quite, it's overlapping, and you can pretty much say that it's correlated by looking at the graph. But just by looking at the graph, can you say that increase in ice cream sales are, causing shark attacks or vice versa? No. So I would like to emphasize here, by looking at any graphs, you can say, okay, two variables are correlated, but you can never conclude by saying that this causes the other one, right? So there are other factors that could involve in this, ice cream sales and shark attack scenario, because both of them happens only during summers, right? So in other words, just because two variables are correlated does not necessarily mean that one causes the other. So one should always consider the other possible factors that may be contributing to the observed relationship, right? Okay, so now that we have covered all the basics of data visualization, let's talk about how we can, let's talk about the role of R in data visualization. Okay. So what is R? R is nothing but a programming language. It's an open source software. It's developed in 1993. And majorly we use R today for statistical analysis, data analysis, visualization, and we also use for certain machine learning exercises, right? So here you can see a graph where it shows the visits to R by industry, and you can see academic and healthcare is one of the top, few of the top industries that uses R. Okay. So R is nothing but a tool, a programming language that we use, that we'll be using for data analysis. And I'll also be showing you certain exercises where we will use it for plotting some graphs. Okay. So what is RStudio? RStudio is nothing but an IDE. IDE is Integrated Development Environment. It's nothing but a platform where you could write code, you can compile it and make graphs, okay? So RStudio will provide you a user-friendly interface. So I'll actually start with showing you this is how RStudio looks, all right? So this is a place where I can write my codes, and the region that you see here is my console, all right? So console is basically the place, the section where I can talk to my computer using programming languages. So let me just check how to. Okay. All right. So this is the place where I can talk to my computer using the programming language. All right. And if you look at the right side pane, like right lower pane here, I have the option to install packages here. Okay. Look for the documentations, and whenever I'm plotting any graphs, I can see it here in this pane. Okay. So on the top here, this is the environment. So you can see all your variables, stored variables here, and you can manage your files here. Okay. So I'm just going to be introducing RStudio, all right, here. So let's start with something very basic. All right. So let me just set up the code. All right. So I'll introduce to something called ggplot. ggplot again is one of the most used data visualization package in R. Okay, so you can install ggplot by going into this particular. Okay, before that, let me also tell you how to install RStudio. Okay, and R. Okay, I hope you can see my screen here. So you can go to, oh, can I move this? Okay, great. This here. So you can go to a website called faucet.co. Okay, go to their downloads and you can see you can install R and install R Studio as well here. All right, so you can choose which OS based on your based on the OS that you use, Windows, Mac, or Ubuntu. You can download the R Studio and run it. Right. So again, you might come across an option where, they'll ask you whether you want the open source or RStudio Pro option. Go with the open source because that, as a beginner to intermediate, you can pretty much do all the functions using open source one. Okay, so once you have installed, this is how it looks like. Okay, so let's try to make something very a small plot. All right. So first, ggplot. Let's install ggplot. How do we install ggplot? Let's go to the packages here. There's an option to install. Click on the install. This small window will pop up and you type what package you need to install. So I'm going to install ggplot2. Okay. Click on the, make sure this tick, make sure here you put a tick so that all the dependencies are installed, and then you click on the install button and it gets installed. This package gets installed. Okay. So other things you need to know here is, install from CRAN. CRAN is nothing but a repository. Our repository is where all our packages are stored. Okay. So I'm not going to be clicking on install because I already have it. Okay. So once you install this particular, okay, right. So once I have installed ggplot, I'm going to call the package. How do I call it? I am going to write a command called library bracket ggplot. Right. So see, there are certain perks of using IDE or platforms like RStudio because it makes your life easier, right? It gives you suggestions. Even if there is some mistake, it will tell you, okay, there is a mistake. I'll show you how it is done. So I'm going to be calling this particular library. I can select this, or I can keep my cursor here and click here, run. Okay. So the other shortcut for run is control and enter. So you can see me doing control enter. So once I have done control enter, you can see in my console, it has run successfully without any error. Okay. I'll show you how error happens. I'm going to remove one G here and I'm going to run it again. So it's going to tell me error in library gplot. There is no package called gplot. Okay. This error, you will get it even if you have not installed the ggplot. Okay. So now I'll go ahead with calling the library. Okay. Second, I'm going to read my data. All right. What is the data that I'm going to use? I have kept some demo data here for the purpose, for our purpose, right? So I'll take something called drug response.csv. This is again nothing but an Excel sheet where I have different doses for drug A and B that I've tested on 2 cell lines, and these are the responses. Okay, so I'm going to be reading this CSV file. How do I read a CSV file? The command would be data. The command would be read dot. See all the options it is throwing up. We'll choose the CSV2, and then I'm going to go with I'm going to go with drug. What was the name of the drug? Response.csv, right? Drug response.csv. Now let's run this. Okay, let's run this and I'll show you the error. Yeah, great. So it says cannot open file drugs response.csv because no file or directory found. You know why? Because I have not set my working directory. Right. So all my files that I'm going to be using, I'm going to use for this particular demo, I have it in my downloads. Okay. So it's better I set my working directory as downloads. How do I do that? I'll go to the files here and I'll click on these 3 dots here. I'll click on downloads. I'll say okay. And now can you see all the, data that I've stored in one place? I'm going to click on this particular small gear here and select set as working directory. It is set. Now let's try this again. Okay, it's running, right? It's just showing me all the information that is there in the file. Now let me store this file by calling it as data equal to, okay, I have stored this file, information in this file, into a variable called data. So if you look at my environment here in the right side, I can see I have a new variable here called data. What is my data? If I click on this one, it will show me the same information that I have loaded from my CSV. Simple. Now I have loaded this data and let me try creating a GG plot. Okay, creating plots using ggplot. Okay, so why did I put a hash here? This is basically to comment out. So all the other lines which doesn't start with a hash, my R will look at it and be like, okay, this is a command, I have to run it. But all the lines that starts with a hash, it will ignore it. Okay, it will know that it's a comment and it doesn't have to run it, right? So whenever it's important that whenever you write a code, you put comments as well, so that later on when you are going back to your code, it's easy for you to understand what you wrote, right? So let's try to create something small like ggplot. Okay. ggplot. Okay. What is the data I'm going to plot? I'm going to plot. I've called it as data. Okay. Comma. AES. So, all right. So AES basically is one of the parameter or one of the parameters that you have to pass in ggplot. Okay, so this is a place where you define the X and Y. All right. So, okay, let me just stop the video. Okay. Right. So AES here, I can define my X axis, which can be dose. Then I'm going to define my y-axis. Let's say I want to see the response in my y-axis. Okay. Now, this is what I want to plot. How do I plot it? Let me look at, I want to plot a scatter plot, let's say. Okay, so I'm going to select geom underscore point. All right, that's it. Let's try running this and let's see what it generates. So do you see what it generates? It generates a scatter plot or a dot plot with y-axis being the response and x-axis being the dose. All right. So now let's try to make it further complicated or further sophisticated. I want to check the treatment also. So I'm going to say color is equal to treatment. Okay, let's plot it again. So do you see how it differentiated the color based on the treatment? So drug A is red and drug B is blue. Okay. Now let's make it further, complicated or further detailed, I must say. Shape is equal to cell line. Cell underscore line. Okay. Let's try to look at it. Yes. So again, it has further subdivided the points into shapes, the triangles being cell line 2 and, the rounds being cell line 1. Okay. So this is a scatter plot. Now, what if I want to add a line on top of it? I'm going to add geom underscore line. Okay. Let's see how it looks. See how we created a graph, which actually tells far more information. It tells about treatment, the line and everything using ggplot. Okay. So now there are several things such as you can give labels. So here you don't have a title, right? You don't have a label. You don't see all those information here. So you can further do that by customizing. So it's important for you to note that ggplot enables, customization of your graph to a great extent. All right. So I'm going to be adding, let's say I want to add labels. Okay, I want to add labs. So this is the parameter I have to write, labs. X is equal to doses, let's say. Okay, and my Y axis, I want to give it as, let's say, response. Okay, so let's run it and check. Yes. So how it just gave me a, okay, let's just wait a second. Sorry. Let me just add a title here. Okay, so how I just defined a title for it, and you can see here, right? I can just customize it however I want. Now let's play around with the theme of it. So generally what happens when you're going for a publication in scientific journals, they have certain rules, right? You can't make it color, you have to give a black background, or you have to do a white background. There are certain restrictions that are there, right? So you can always choose a theme to your graph. So let's look at some themes that is available in ggplot, right? So when you click on theme underscore, you will have a lot of options. Let's try the first one, BW. It is giving you a, okay, so this one is what we have default shows. Let's look at classic. Okay. So I think even in this one, the background has changed, right? So even in the classic, it would change. It would remove all the extra, yeah, it has nothing in the background, right? So there are multiple themes that you can play around with here. So the point is, it is highly customizable, and ggplot, you should check its documentation. So whenever you want to look at, documentation for any package, you can go to our documentation and you can look for the documents. Right. So here I have the documentation for ggplot2. So when you click on this particular ggplot2, you can see what are the parameters that it needs to use, and you can further go on to understanding how to use, and all the information would be there in its documentation. All right. So let's look at one more example. So here we created a scatter plot, right? Scatter plot using a scatter plot plus a line chart. Now let's try to make a bar chart. Okay. So for bar chart, again, I have another CSV called bacteria.csv, where I have sample A, B, C, and I have tried looking at the abundance of these bacteria, 1, 2, 3, 4 bacteria in all these samples. Okay. So how do I go ahead? First is reading the data. So I'll do it a little more faster this time because I've already introduced you to the basics. So let's call it as back data is equal to read.csv, and inside, what was it called? Bacteria. I think somebody's mic is on. Okay. Okay, thank you. I'll share the screen again. Right. So I'm going to be reading the data. Okay, this time my data looks like this. And let's just move forward with creating the ggplot. Okay, and I'm going to go data AES bracket X is equal to sample, Y is equal to the value. So where is my, just give me a second. Let me just have a look at the, sorry. My X is the sample and my Y is the abundance, right? Abundance, yes. I'll go with the abundance. Abundance. And let's try looking at geom_bar. So again, I want you to just look at all the options. So you have area, chart, bar, chart, box, plot. Okay, there are so many options here. So we'll go with the bar chart here. All right. And inside, I'm going to write stat equal to identity. Let's try to. Oh, okay. So it's just giving me sample A, sample B, sample C, right? And it's abundance, it's 100% only, but I have no information of my bacteria here, right? So what is my bacteria? Bacteria column, I will do, yeah. So my X is sample, Y is abundance. Let me fill it with bacteria, okay? Now you can see how it made the difference. Now let's look at it. This is a stat bar chart. For sample A, my 25 a bit, 30% maybe is bacteria 4. So in this way, I have filled the bar charts using multiple colors, right? Based on the information of the bacteria, right? This is again simple. So these are some of the two simplest examples that I could give for using ggplot, right? So it's not limited to these type just because of the time constriction. I have chose to just give you these two examples, right? So now let's go a little more further in detail, okay? So whatever you just saw right now are some of the simplest normal graphs that you can use for any type of data. However, in the context of biology, we researchers have way more complex and large data, and then we use certain graphs like heat map, phylogenetic tree. These are some of the graphs that are specific to biological data, right? So I'll be giving you, a brief or just an introduction to how we can use R for making a heat map or making a phylogenetic tree. Okay, so let's look at it in the next 10 minutes. Again, I want to say this webinar is for beginners, absolute beginners. So for the next 10 minutes, even if you're not able to understand many concepts, it is okay, because I'm just going to introduce you to some, deeper concepts. Okay, so let's look at the R Studio. Okay. Right. Okay. Let's look at. Okay. So let's say I have a gene expression data. Okay. I'll show you my gene expression data. Okay. Before that, let me just show you how to clear my console, clear the variables, to avoid confusions. Okay, so do you see a small broom icon here? If I click on this one, it will remove all the variables that I've stored, right? Earlier, I had saved something called data. It's gone now. Similarly, I can, click on this broom here and then click yes. All the history of my plots will also be gone. Similar is available even here. I can clear everything and start afresh. Okay, so here when I'm looking, I'll show you a gene expression data. Okay, this is my file. Yeah, gene expression data. Let me read the gene expression data and I will show you what it is. So here I have 10 genes, or 20 genes and 10 samples, and these are all the expression levels. Okay, and what I'm going to do, what I'm going to do is, first I'm going to read the data. Second, I'm going to extract only the numerical value. Okay, how do I extract the numerical value? So let me look at the gene expression data. So this information here, only this information. Ignore the first row, first column. This information alone is my numerical data. Okay, how do I do it? I can use this. So square bracket is used for slicing the data. Okay, so I'll not be able to go much in detail today, but this is how I'll be slicing the data and taking only the numerical data. Let's read on this numerical data. Okay, great. Now that I have the numerical data, let me just check if I have read it properly or not. Okay. Gene expression matrix. If I click enter in the console, it should just show me what the data contains, right? So here it is. Now I'm going to use an inbuilt function called heat map for creating or generating the heat map. Okay, so I didn't do anything. I just took the gene expression data. I made it into a matrix where I can just take the numerical values and then I use the heat map. And here you see I've made the heat map. Again, you can ignore these dendrograms and all the other details. But here I have found a correlation. So things like, sample 9 and gene 4, there is a darker color. That means maybe a higher correlation, right? So this is very simple. Now, again, I can customize it. Okay. So you have a package called RColorBrewer. You can call this particular library and you can use the same heat map function. However, in the color section, I can give a certain code, right? So again, I can enter it and see how it changed, right? So the color combination is changed. I have the control over it. Okay. Now I don't have a legend, so I can add a legend here by adding this chunk of code. Let's not focus much on these. These are just to show you how much customizable these R visualization packages are. Let's not break our head onto the details of what code does what. Okay. For now, at least, right? There's one more part that I wanted to cover today is about phylogenetic analysis, okay? And bioconductors, basically. So seq-in-R, APE, these are all some of the, packages, R packages that is used for reading FASTA. So seq-in-R is used for reading FASTA files and APE is used for phylogenetic analysis. MSA is used for multiple sequence alignment, okay, which is a part of phylogenetic analysis. Now let's try to look for installing MSA. Okay, so I'm going to type MSA here and I see there is no MSA here. Why? Because MSA cannot be downloaded from CRAN. Basically, it's not a, so this is special package that is present in something called bioconductor. Bioconductor is a repository where you have all the tools or all the packages that is required for genomic data analysis stored in one place. Okay, so maybe I can show you how the site and I can show you how to install it as well. Okay, let's look at this. Okay, so bioconductor here. So this is the site. You can go to bioconductor.org and try to install. So you can install the bioconductor by using this code, right? Once you have installed it, you can go to bioconductor and search for MSA, right? So this is the MSA tool that we are going to be using. You copy this entire chunk, copy it, and you know where to put it. You can put it in your RStudio, right? You can go to your RStudio in the console here and you can paste it and enter it. Okay, so it will install the MSA package. So again, so this is the MSA package, right? So now let's try to make, I have made a small FASTA file which contains angiotensin, the hormone and angiotensin and insulin's information from multiple species. Okay, so I'm going to be reading the FASTA. Just like how we read the CSV file for FASTA, we have a different code, which is read a string set. So I'm going to be reading it, right? Oh, sorry. I have to first load the packages seq-in-R, APE, and MSA. Okay. So once all these packages are loaded, so it's taking some time to load. Okay, it is done. So once all these packages are loaded, I can run this. Okay, now can I see what my sequence is? I'll just type sequence. So do you see how it has read the FASTA file here? Okay, so I have 4 sequences here. All right. Now I'm going to perform the multiple sequence alignment here by just adding a small code. I'm going to give the sequence as an input. Okay, so let's run this. So it would take some time if you are providing a larger FASTA file, but because it's a small ones, it didn't take much time. So again, one part of phylogenetic analysis is calculating the distance matrix. Okay, again, we'll not be going much into detail in here. I'll just be running it to show it to you how simple it is. Okay, I'll just run this. All right, so my alignment is done. So let me just check alignment, right? My alignment. So it uses something called cluster W. It'll try to match. These are like some core concepts of bioinformatics. Again, I'll not be going into detail. So once we have calculated the matrix, I'm going to make the analysis. I'm going to do the analysis and plot the tree. Okay, so this is basically my tree. Okay, so okay, further, I think I have some information in my slide with respect to this particular phylogenetic tree. Okay, let me just show my slide again. Okay. Okay, so this is the tree that we just made. Okay, so angiotensin receptor for 2 different species of rat are clubbed together, are put together. Similarly, another rat's insulin protein or insulin FASTA DNA I've taken, that particular thing is being classified as separately and does not belong to this cluster, right? And again, insulin from Aplysia californica it's a sea slug basically. It's again put into a separate branch. So you see how it just calculated the multiple sequence analysis, calculated the distance and plotted the graph, plotted the tree quickly, right? So again, I'll not be able to talk much about what these branches are, how the matrix is calculated, and what are the basis of bioinformatics behind the multiple sequence alignment. So if you are interested, it is worth, you can check out some of the modules given by Nyberman itself. So you have one module for module 5 on R for biologists. So here we'll be covering much more details. So you will know what the Z-score in a heat map stands for, right? I will be, we'll be teaching you the basics of R, starting from installing and setting up your workspace to the complete NGS data analysis, right? So if you're interested, you could look for this particular module here in Nyberman. So maybe I can request somebody from the team to put the link, necessary link, so the students can access it. Okay. So overall, in today's webinar, we looked at the basics of data visualization, and then we also looked at how easy it is to plot graphs in R, right? However, due to time constraints, I was not able to go in detail with respect to the biological data analysis and visualization part. Like I mentioned, you can check out the courses, and whoever is interested can apply for it. All right. So thank you so much for the coordination. Now I'll be taking questions. Let me, there's a lot of questions. What I can probably do is, our team has curated few questions, and I could read from there and I can answer it, right? So can you, so you have a question from Sukanda Devnath. Can you describe the steps of data visualization? Okay. So steps of data visualization, simple. You take the data set, it starts from the data set, you do the processing, right? You do a lot of data cleaning as well. Sometimes what happens that when you have a lot of outliers or data that is not worth, plotting, you could clean the data and bring everything into the same scale and then plot it. Plotting again, before plotting, you need to decide what chart to use, based on the slides that I have mentioned. And then further you can go ahead with choosing the right packages in R for plotting. Okay. You have a question from Sheikh Nizamuddin. How to specify my own color and shape in ggplot? Okay. So you can actually choose your colors. You can go to some website, find the hex code. Hex code is the code that is used for identifying colors, right? So every color has a specific hex code, right? So you can use that hex code, put it in the appropriate pass it through the appropriate parameters in ggplot. Again, it's worth to go through the ggplot documentation onto where you want to put it. Okay. Then okay. Let me just go through the questions. I think that's it. I guess there's a lot of conversation going on here in the chat box. I can't really are there ways to plot graphs on R without the use of codes? Unfortunately, no. I mean, you can use other tools, right? There are several tools where you can upload your data and then they will generate a map for you or will generate a graph for you. But if you want to use the R Studio and R, you need to have a basic knowledge on R, right? Again, it's not a very difficult language to learn. You could use, you could easily learn it in a span of few weeks, okay, if you really work harder, right? Then can you share this R Studio link? Yes, I can. So there are chances that I might not be able to, answer all of your questions. What you could probably do is post the same questions in our forum. And I will try, answering you guys there. And I can also provide the necessary link and reading materials there as well. Okay. And okay. I think we have run, I think it's time is also over, right? Change the fonts. How to change the fonts and question from Datta. How to change the fonts and font sizes in ggplot? Because when you export a plot to RStudio, the fonts tend to be small. A very, very good question. And I should also emphasize on how to save your graph, right? So let's say this earlier graph, let me just run it again for the sake of it. So I have this particular graph here. You can change the size of the, what do you say, columns or the fonts by passing certain parameters. Again, I would suggest you to go look at the documentation. In this example, I can show you the sex 0.6, right? So 0.8, let me just put 0.4. Okay, I'll just add it. So see how it reduced okay, it overlapped, but you can see how it reduced the legend, right? The size of the legend here just became smaller. Similarly, every font can be controlled, right? And how do I save a particular graph here? I can click on export. I can save it as a PDF or as an image. Click on the image. I want to save it as an image. I can select the width and height. Okay, I can select the format and I can click on save button here. Okay, so this is how you can save the graph in ggplot. I think okay, this what's the best? Okay, so you have another question. Can you give us a way to practice R in data analysis, as it is the best way to learn R? Where to find exercises with some real data? That's a very good question. Thank you so much for asking. So you can check out several websites. So you have Kaggle. Okay. So Kaggle, again, is a website where you can find real life data. You download the data sets from there and you can move forward with learning how to do the analysis. There are today there are a lot of tutorials online, and it would be easy, but about finding the real life data, you can go. Kaggle is a very good source. Okay. So I think the remaining questions you could put in the forum, because I'm getting the message from the team that the time is over. So I would end the webinar here. Thank you so much for attending the webinar, and I really hope you guys learned something new today, and looking forward for further, more webinars like this. Thank you.